Hello, everyone, and welcome to today's webinar, Driving Protein Research, Automated Liquid Handling, and qPCR for Extraction and Thermal Analysis. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Analytic Yena. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I do also want to let you know that we will have a brief survey after the webinar, and we would appreciate your participation in this survey. I'd like to now welcome our speakers for today's webinar. Jason Dexter, Application and Commercial Specialist at Analytic Yena, and Matthias Ludwig, Application Specialist at Analytic Yena. Jason, I will now pass it over to you to begin your portion of the presentation.
Hello, and welcome to our webinar for Driving Protein Research with Automation and qPCR. Myself and Matthias will be talking through Analytic Yena's solutions for automated extraction and thermal analysis. Protein-based therapeutics represent a significant and growing proportion of drug development within pharma R&D. Protein-based drugs likely represent over 30 to 40% of current pharmaceutical R&D pipelines. These include proteins such as monoclonal antibodies and antibody-drug conjugates used in oncology and autoimmune diseases, enzymes for enzyme replacement therapies, as well as hormones and vaccines. Because proteins are complex molecules, they require careful study during drug discovery and development. They are powerful and highly specific, but are sensitive to the environment that they're in, causing issues such as aggregation and degradation. This requires stability testing at low and high volumes, as well as reliable characterization to ensure quality and regulatory approval. Protein purification is the first essential step after expression. This removes host proteins, DNA, endotoxins, and other contaminants for downstream applications. Only pure proteins can be reliably analyzed for functionality, stability, and reproducibility of screening assays. If you have poor protein purification, this can lead to failed experiments, wasted samples, and higher development costs. As part of the protein characterization workflows, Analytic Yena has worked closely with customers to deliver an end-to-end approach, ensuring high-quality protein purification as well as analysis. Our solution for high-throughput automated protein purification is to take advantage of the flexibility of the SciBio Felix automated liquid handler and to use the tip-based technology for simple and fast purifications, and to characterize your purified proteins using the real-time PCR thermal cycler qTower for the thermal shift assay. Using the SciBio Felix, you can choose between one of two different tip technologies, Biotage's PhyTips and IMCS tips. The basic principle is the same across both brands, however there are some slight differences between the two. They both make use of 1,000 microliter tips that include a commercially available resin. For PhyTips, this resin is densely packed in between two screens, and for IMCS tips, it's loosely packed within the tip. IMCS also have this patented disperser within the tip that allows for turbulent mixing of the resin and the sample to maximize contact time and binding potential. So why use a tip-based purification method? Sure, there are other chemistries out there such as magnetic bead separation, but for tip-based purification you require less lab wear on deck and no chance of beads left over in your sample. Resins within the tip can be changed based on your desired proteins, and Analytic Yena have also automated an IMCS buffer exchange method using their size X tips. A full plate of 96 samples can also be purified in under an hour. As I mentioned, the tips utilize commercially available resin. So if you wanted to purify antibodies, you would use a protein A resin. For histag proteins, it would be a nickel iMac, and for biotin-related proteins, a streptavidin resin. When ordering tips from each supplier, you can choose between several different resin volumes, ranging from 10 microliters to 100. This would be determined by your workflow and your desired protein concentration after purification. The higher the resin volume, the more protein you are able to purify. Here is some data generated by IMCS. They were purifying histag GFP using two different resin volumes, 25 and 50 microliter nickel iMac resins, on the SciBio Felix. It shows binding cycles, which is the amount of times that you aspirate and dispense your sample over the resin compared to the amount of protein bound using known different starting concentrations. Within the 25 microliter resins within the tip here on the graph on the left, it shows that if you started with a concentration of 1 mg per mil, to get 1 mg per mil of your protein back, you would need to have 20 binding cycles. Compared to if you doubled the amount of resin within the 50 microliter, the graph on the right, to get the same yield, you'd only need 10 binding cycles. The difference in binding capacity within the different tips would allow you to speed up your process while still getting purified protein back. So let's quickly talk about the biofelix. It is a compact benchtop liquid handler with interchangeable heads, giving you the ability to go from pipetting 96 to 384 samples at once, and an overall volume range of 1 microliter to 1 ml. The protein purification workflow uses the R96-1000 head, and here you can see how the 12 deck spaces are laid out for this workflow. The phi tips, or IMCS tips, are loaded into the SciBio Robo tip trays and placed on deck along with the chosen labware before running the composer script. So here are some of the variables within the scripts that allow you for a bespoke tip-based protein purification workflow. As mentioned before, your chosen resin dependent on the desired protein, as well as the resin volume determined by the final protein concentration. There are workflow components such as optional pre-filling of plates, as well as potential neutralizations of after elution for workflows that use protein A resins. Sample count, where you need to do a full plate of 96 samples or a specific number of samples between 1 and 96. Labware can be customized to what you have on hand, as well as the number of wash and elution plates, the number of equilibration cycles, sample binding, as well as wash and elution steps. These are all changeable within the script. They are also available in both SciBio Composer scripting software if you need to make any changes, or if you want to change some specific variables, then the scripts can be run in our Application Studio software. The scripts can be pre-written methods that are ready to use for standard workflows, or customized methods for optimized processes. Here we have a video of the IMCS workflow purifying his-tagged GFP using their nickel iMac tips. It's worth noting that the steps would be very similar, if not the same, compared to the phi tips. The first step is to equilibrate the resin within the tip. Next is your sample binding. Here you can see the resin becoming brighter and brighter as more of the GFP is becoming bound to the resin. Interestingly, you can see the resin move within the tip up and down during the binding cycles. If you were using phi tips, you would still see the resin become brighter, but as the resin is packed in between the two screens, it wouldn't move within the tip. We then move on to our first of our wash steps. These wash the resins and help remove the contaminants. The number of wash steps is variable within the script and can be optimized for your workflow, but here we are showing 2 wash steps. We then move on to our protein elution. The elution buffer allows the proteins to become unbound to the resin, leaving you with contaminant-free, high-yield purified proteins. Finally, we have some data from our application notes automated phytic protein purification on the biofelix. In triplicate, 1 ml of bacterial expression culture was grown for 16 hours. The his-tagged proteins were then captured using phi tips, nickel iMac tips, using both 10 and 160 microliter resin volumes. The proteins were eluted in 2 separate successive elution steps of 100 microliter of elution buffer. The protein concentration was then measured using the Bradford assay. The yield and final concentration of proteins recovered correlates with the quantity of the phi tip resin used for the purification. Additional purified proteins can be recovered by increasing the elution volume as desired. The purity of the purification was then assessed by SDS page, where a band was isolated at the expected weight of 28 kilodaltons. A likely isoform was also identified, which was also present within the bacterial isolate. The other bands were likely E. coli proteins with native histidine residues commonly seen within the his-tagged protein purification methods. Automation of tip-based purification with the biofelix is the ideal solution for high-throughput protein purification in a compact and powerful format. Pre-written methods are available for the biofelix to perform purifications in an accessible and flexible format. After your proteins have been purified, your downstream workflows are going to further characterize and test your proteins. Matthias is now going to talk about the protein stability analysis using qPCR instruments.
Thank you, Jason, for this overview about automated protein purification. Now I would like to talk about protein stability analysis using qPCR instruments, like our qTower Iris. Protein stability is a key in the drug discovery, structure biology, and formulation. It's used to investigate protein folding and unfolding, biomolecular interactions such as protein-protein, protein DNA, or protein lipid binding. There are two common methods on the market or in the research area. This is TSA and DSF. These are methods to determine the thermal stability of proteins. DSF stands for differential scanning fluorimetry. It's like an umbrella term for temperature-dependent fluorescence monitoring. And TSA, the thermal shift assay, is a classical approach using dyes to monitor unfolding and ligand binding. I would like to have the focus on the thermal shift assay or TSA. Here you can see the main principle of the TSA. Thermal shift assays are commonly used to study protein interactions and stability. By gradually increasing the temperature, proteins begin to unfold, exposing hydrophobic regions. Fluorescence dyes, such as cyper-orange, bind on these newly exposed regions, resulting in a change in fluorescence intensity. This signal is tracked in real time to generate a melting curve, from which the protein's melting temperature, Tm, can be determined. This is a key indicator of structural stability or binding events. This process is comparable to the qPCR melting curve analysis with cybergreen. Depend on your protein are different dyes available. For example, for soluble proteins, we have established dyes such as cyper-orange or glow melt, and also ANS. ANS reports on exposed hydrophobic regions, works also for some membrane proteins. These dyes are easy to use, highly sensitive, and also compatible with standard qPCR optics. But for membrane proteins, you need some specific dyes, such as CPM. This detects on the cysteine exposure during the unfolding, and NIRAD. That monitors hydrophobic lipid environmental changes. These dyes enable reliable stability assays in detergent or lipid carriers. For all of these dyes, it is important that you check the emission and the filters on your qPCR device. CPM cannot be directly measured on a standard qPCR instrument. Specialized equipment with UVA excitation is required. When it comes to performing thermal shift assays, several instrument platforms are available, each with its own strengths and limitations. Plate readers are widely accessible and offer flexibility in experimental setup, but they are often limited in temperature precision, which can affect the resolution of melting curves. NanoDSF instruments, on the other hand, are label-free and highly sensitive, providing detailed information on protein stability. But they come with a high cost and may not be suitable for high-throughput screening. qPCR instruments, such as the qTower series, provide a practical balance between these approaches. They offer highly precise temperature control combined with a real-time fluorescence detection, enabling accurate, reproducible thermal melt measurements. This makes them particularly well-suited for TSA applications, allowing researchers to efficiently assess protein stability, screen multiple conditions, and work with minimal sample volumes. So, why choose qPCR instruments for thermal shift assays? Building on what we have seen about plate readers and nanoDSF, qPCR devices combine the best of both worlds. They are fully compatible with standard fluorescence dyes such as CypherOrange, ANS and CPM, and allowing flexible assay design. They provide high precision and reproducibility with the added ability to monitor fluorescent signals in parallel across multiple wells, which formats like the 384 well block qPCR instruments enable through high-throughput screening while keeping sample volumes minimal. And importantly, they offer a cost-effective alternative to more specialized platforms like nanoDSF. Now that we have covered the principles, dyes and instrumentations, let's move on to a practical step. In the following sections, we'll talk through a step-by-step TSA workflow on our qPCR cyclers. We'll demonstrate the process using CypherOrange and ANS for soluble proteins and CPM for membrane proteins, including an example protocol and representative results. CypherOrange is a fluorescence dye that binds on the hydrophobic regions exposed when proteins unfold. It has been widely used for thermal shift assays due to its versatility and reproducibility. The dye is fully compatible with qPCR instruments, enabling high-throughput analysis of multiple samples and conditions. It works particularly well for soluble proteins and allows researchers to efficiently compare protein stability across different buffers, ligands, or variants. Here you can see our experimental setting for the CypherOrange thermal shift assay. We used a sample alpha-chemotrypsinogen, that's a soluble protein. We performed a thermal shift assay with CypherOrange and different PBS buffers on different pH values, like pH 5, 7, and 9. Our qPCR device was in qTower Iris 384, inclusive the protein module number 1, with the wavelengths 465 and 580. And we make the analysis with the melting curve tool. Here you can see the results of the melting curves of alpha-chemotrypsinogen in PBS buffers of different pH values. Here on the left side, these are the monitoring curves of the melting curves. The red line is for pH 5, the green for pH 7, and the blue for pH 9. And then we would like to have the first derivative of melting curve to get the melting temperature, to generate peaks at the temperature with the highest change of fluorescence. For this, you have to flip the curve, because in different to a standard melting curve experiment with cybergreen, we have not a decreasing of the fluorescent signal, we have an increasing of the fluorescent signal. This is the main difference to a classical melting curve experiment. In summary, you can see here the single melting temperatures for the depends on the different pH values, and also the mean melting temperature of the pH values. The fluorescence intensity of CypherOrange was inversely affected by the pH of the buffer. Highest fluorescence intensity shown at the low pH, and the highest protein stability also at a low pH 5. Consistent with the fact that alpha-chemotrypsinogen is a physiological present in a low pH environments. As a second example, I would like to show you the results with the other dye, such as 1,8-ANS. We used again alpha-chemotrypsinogen, but now with different protein concentrations in different volumes. For the qPCR assay, we use this 1,8-ANS, and you can see here the different excitations and emission wavelengths of this dye. We use different protein concentration and also different volumes. For our qPCR one, we use as a equipment the qPCR Iris series, inclusive the color module number 7 for UVA, and also the color module number 1. This is then our blue channel. The analysis tool is the same as for Cypro Orange, and for the others, we use the melting curve analysis tool. ANS is a fluorescence dye that binds more weakly to proteins than other dyes, making it highly sensitive to subtle conformational changes. Upon binding to low hydrophobic environments, it exhibits a strong increase in quantum yield, providing a clear fluorescent signal. ANS is versatile and suitable for soluble and membrane protein studies. It's a low sample requirement down to 5 microliters per well in the 384 well plates, and makes it practical for screening multiple conditions efficiently. Now we come to our third dye, CPM. CPM is commonly used to assess the stability and solubility of membrane proteins, which is especially important for purification and downstream characterization, such as crystallization. The dye is a non-fluorescence when unbound by the fluorescence upon binding to the cysteine residues in the intermembrane regions. During the temperature-induced unfolding, previously buried cysteines become exposed, allowing the fluorescent signal to report on the protein integrity. The fluorescence intensity also increases with protein concentration, providing a sensitive measure of stability and folding. Here you can see the results of our CPM experiment. We use as a model protein beta-lactoglobulin in different pH values. Our qPCR device was the qTower Iris 384 UV, inclusive the color module 7 for the UVA range. The CPM-based TSA can be performed also in a 384 well UV compatible PCR setup. And we checked not only the influence of the different pH values, we checked also different reaction volumes. For example, 10 microliters and 20 microliters. The fluorescence intensity is lower at 10 microliters compared to the 20 microliters. These are here the lower curves. But the melting temperature remains unaffected by the change in the reaction volume. And so it is suitable for assessing protein stability in a small volume and in the high throughput experiments. So that you can perform also a good thermal shift assay, I would like to give you some information about qPCR based TSA. Sample preparation. A proper sample separation is essential for reliable thermal shift assays. Use gentle buffers, avoid tetogens when working with Cypro Orange, and reducing agents when using CPM. Optimize dye concentration to achieve a good balance between signal and background. Keep samples on ice or on cold block before measurement, and work efficiently to minimize protein degradation. When you handle with the dyes, fluorescence dyes are light sensitive, so handle them carefully. Use dark reaction tubes and seal plates tightly. For dyes such as CPM, work in a dim or darkened room and protect samples from direct light to prevent bleaching. Prepare dyes mixes freshly before each experiment to ensure consistent results. About the qPCR specific workflow. To obtain well-resolved melting curves, pipette gently to avoid bubbles and seal plates tightly. Make sure to select the correct fluorescence channel for each dye. Use low temperature ramps, 0.5 to 1 degrees per minute, to improve curve resolution. For reliable melting temperature values, perform measurements in triplicates whenever possible. To summarize, using qPCR instruments for thermal shift assays offers multiple key advantages. First, dye compatibility is broad. Cypro Orange and ANS are excellent for soluble proteins, while CPM can be used for membrane proteins. Although CPM requires UVA excitation, so a standard qPCR channel won't detect it directly. Second, qPCR provides high precision and reproducibility. The temperature control is accurate, and a parallel detection in multi-well plates reduces variability between replicates. Third, it's highly cost and time efficient. By using existing qPCR devices, labs don't need specialized TSA instruments. Small sample volumes save protein, and the 384 well format allows high throughput screening of multiple conditions as a single one. Finally, qPCR-based TSA is easy to integrate into routine workflows. It's compatible with standard buffers and microplates, and it can even be adapted to automated or semi-automated setups. In short, qPCR enables sensitive, reproducible, and high-throughput protein stability analysis without the high costs of dedicated TSA instruments. In summary, this webinar highlighted how automated protein purification and thermal shift assays, TSA, using qPCR instruments can accelerate and simplify protein research. For protein extraction and purification, tip-based technologies were presented as a space-saving and flexible solution. They streamline workflows, reduce hands-on time, and enable rapid buffer exchange. With these systems, up to 96 samples can be processed in parallel in under 1 hour, ensuring efficiency and reproducibility. For protein stability analysis, TSA on qPCR instruments was introduced as an affordable and powerful alternative to other thermal analysis methods. This approach combines enhanced sensitivity and high throughput with minimal sample consumption. Using 384 well formats, hundreds of samples can be analyzed in a single one, making it ideal for large-scale screening and comparison of protein variants, buffer conditions, or lichens. Together, these methods provide an integrated workflow from purification to characterization, supporting faster and more efficient protein research. That concludes our presentation. We welcome any questions you may have.
Thank you for your informative presentations. Joining us for the Q&A session is Anna. She's an application specialist in the liquid handling department at Analytic Jena. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Q&A box below and click submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question is for Matthias. This question is asking, are specialized low bind plates or tubes recommended when performing thermal shift analysis with Cypro Orange, ANS, or CPM?
That's a good question because low bind plates are very popular in protein workflows, but we could see no effect on the thermal shift assays. And so you can use in principle also your standard PCR plates or qPCR plates that you have in your lab for such kind of thermal shift assays, independent of the dye. Is it Cypro Orange, CPM, or whatever? The only recommendation is, well, this was part of our web seminar, that you work fast and maybe in a dimmed room, and you use maybe white wells for your qPCR and your tubes for pipetting. It's the best way if they are in a dark colored. Yeah, but special low bind plates or tubes are not necessary.
Excellent, thank you. Okay, our next question I'll hand over to you, Anna. This question is asking, what yields would you expect with the different resin volumes?
So we have limited data for the phytips with regards to the yields. I can say that for the iMac, tips from Biotage, so the phyte tips. If you use 10 microliter of resin, you have a yield of about 200 microgram of protein. The same is true for the IMCS tips, and here we also have some data for larger resin volumes. I think, if I recall correctly, there's yields possible up to 1 milligram of protein for the 250 microliter resin volumes. And if you go higher, then you can get yields of more than 1 milligram of protein as well.
Excellent. Thank you. Okay, our next question, back over to you, Matthias. This question is asking, can these dyes be used to accurately determine protein concentration in a qPCR-based thermal shift assay?
Not directly, no. You can see, yes, I have shown this also in my presentation on my part. Yes, you can see an effect if you have for a higher protein concentration and you work with ANS or CPM, then you have also a higher fluorescent signal. But it's not direct linear to the concentration, and it's also not so easy to create a calibration curve to determine the correct concentration of the protein. But in principle, yes, it's possible to see a difference in the concentration, but not in a direct relation, so that you say, okay, this is this milligram per milliliter or this milligram per milliliter. This is not possible with such analysis tools like melting curve analysis also. You need a specific tool, but we could not see an indirect effect. Yeah.
Thank you. And we have another question that might be a little bit related, so I'll keep this on you, Matthias. This one is, to what extent can thermal shift analysis differentiate between proteins with similar structures using these dyes?
No. You have to know what protein you have in your plate or on your tube. It's not possible to distinguish with this different dyes between different structures or different proteins. No, you have to know what is your protein and about the structure. You have to know this. Yeah.
Okay, thank you. Anna, over to you with this next question. This question is asking, what is the maximum sample volume that you're able to purify? Your microphone is muted. I'm so sorry, Anna.
Sorry. Okay, so the Felix in general is able to move volumes of up to 1 milliliter. And phyte tips as well as IMCS tips are available in this size as well. So they are 1 milliliter tips. So that means in one pipetting step, you can take up 1 milliliter of sample. But of course, if you have larger sample volumes, you could distribute those across several wells or even several sample plates, so that you can address larger volumes by multiple binding steps. So you would move with the same tip to the next well that contains the same sample, and thereby you could process, kind of indefinitely large sample numbers. Of course, this would draw out this whole process quite a bit. But if you know that you have like a low expression protein in your sample, so you're expecting small amounts of proteins in a large amount of volume, then you can reduce binding cycles, for example, because it won't take that long for this little amount of protein to bind. So you could move through a large volume, but still reduce the time by reducing binding cycles and still get a lot of yield out of your sample.
Excellent. Thank you so much. And it looks like we have one more question here today. So if there's any additional questions, please feel free to submit them and we will try to get to them today. This last question here for you, Matthias, this one is asking, is the protein melting thermal shift assay also usable for stability research of protein complexes, or will you get two peaks in the melting curves?
We use here in our lab only some model proteins. It could be possible that you can use it, but in principle, yes, if you get two peaks, for example, in melting curves, then it gives you the idea or the information, okay, I have not maybe in my sample or in my well one protein, or I have a complex, so that I have an unfolding in two different temperatures. And so it gives in principle you the information. It could be a complex of different proteins or different structures. But we never tried this. Maybe it's a good idea to check, is it possible to use this principle also for protein complexes. But in principle, when you get two different melting peaks, then this is also an information that you have maybe two different proteins or different stabilities at the same conditions in your sample. That could be possible. But we never tried this. So we have not the experience about such protein complexes.
Excellent, thank you. Okay, we had another question come in. This question is asking, how can real-time ultra-fast measurements of protein conformational changes, e.g. using fast deadroom lab techniques, be integrated with molecular dynamics simulations to reveal the complete folding energy landscape of a protein?
It's a very fast experiment. In principle, we try to reduce the scanning times and also the ramping rates. So in principle, the fastest what we performed are a complete from 30 degrees to 95 degrees was around about 15 minutes. So this is but we could see an effect also when it's too fast. Then you have not enough time, or the dye have not enough time to bind. Also, you have to give this process also time, and also you have to give them the temperature or the block time to heat up the sample in your well. Yes, when you work with a 384 well format and with low sample volume, for example, like 5 microliters, then it's also possible that you can reduce this and makes it faster to see maybe also an effect on this. But what we did here is we checked in principle often with some model proteins and so some typical applications. But we are totally open if you would like to try this or to optimize this. We are very, very interested in to collect or to help you to make your research more efficient. And because we offer only the tools to make such kind of protein stability tests. But we have not the possibility to do everything. But if you would like to do this, maybe we can arrange an contact, and so that we can adjust this for your application, so that we can make such kind of ultra fast measurements. And to see an effect, for example.
Excellent, thank you. And it looks like that is all of the questions that we have for today. So I'd like to thank you for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Analytic Hyena, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And I'd like to remind you at this time that there will be a survey popping up before you leave the page. And we appreciate your feedback. Until next time, take care, everyone. Goodbye.
Bye-bye.
Bye-bye. Thank you.